All right, I think we'll get started. So it's my pleasure to welcome Maeve. Maeve is our newest biology hire. Her second semester here, and we're very excited to hear what she has to say. All right, take it away.
So as Cam said, yes, I am Maeve. I am assistant professor in biology, new professor, and I mostly study bacteria and phage. So we'll talk a bit about that today and a bit about some of the stuff we're going to be doing in some classes. Bacteria, they're everywhere, they're awesome, and if you think otherwise, you're wrong. That's fine, right? They do a lot of different really cool stuff. They're some of the best biochemists around, right? They do a lot of really weird biochemistry in the ground to make a lot of weird compounds. They do a lot of carbon fixation, right? They do a lot of photosynthesis. But one of the things that I think is super cool about them is the sheer abundance of them. So in total, we estimate that there's about 10 to the 30th bacteria on the planet, right? Which is an enormous number. And for reference, we estimate that there's about 10 to the 25th stars in the universe. So there's about 5 orders of magnitude more bacteria than there are stars, right? But these are actually only the second most abundant organism on the planet. The first is actually phage. So phage are these little viruses that particularly infect bacteria, okay? They do not infect humans in any way. They don't infect most animals. They only infect bacteria, right? And they're about 10 times as abundant as bacteria, right? And these are not just simple little organisms. They don't just float around. They're not just bags of goo. They're not unorganized. They're actually incredibly complicated, right? And they're able to do a lot of different really cool behaviors, right? So this is all stuff I used to work on in grad school, right? So this is Myxococcus xanthus. Myxo is a predator, right? So it actually eats other microbes for nutrients, right? It doesn't really like sugars much, right? So on the left here is a video from Jeff Berleman. This is a Myxo colony, and this is E. coli. When we start it, you see that Myxo will swarm out and basically just devour that E. coli colony, right? This is very sped up, I will say. This is not as fast as they move. They actually only move at half the speed of tectonic plates. They're very, very slow, right? But if we speed it up, it looks really cool, right? But so let's say what happens when they stop having prey around? Well, they can then go into this multicellular developmental life cycle where they form these little fruiting bodies, right? Which referred to as these little mounds, right? We get this terminology actually from fungi and mushrooms, right? And this is actually because for a long time, Myxo was mistaken as a mushroom because we didn't know better, right? And so they make these little mounds that contain about 1 million cells apiece. And this is a really coordinated multicellular effort. And in grad school, I used to study how this happened. So this video on the upper right is a video showing some cells as they start to mound together into these really amazing structures. And weirdly enough, sometimes these mounds are able to actually move. And these are millions of cells at a time, right? And then they go through this developmental life cycle. Right, so they're able to do really complicated, really cool behaviors. But the question is, how do they control this, right? You don't want to be doing these all the time, right? You don't want to be developing while you're trying to eat. How do you choose one or the other, right? And the thing that I studied to understand this is what's known as transcription, right? So this is the ability essentially to turn genes on or off, right? Think about this in terms of like a light switch or your house, right? Let's say you walked into your house and all the lights turned on. That would not be super efficient. You're not in all your rooms. You don't need the light on in all your rooms. It's wasting a lot of energy. The other thing is it can be pretty deleterious for you. If you were in your house and your lights were all always on, how would you go to sleep, right? So you want to be able to control when this is turned on and when this is turned off and able to essentially function normally. And this is sort of what gene expression and transcription controls, right? It controls when these things are on and off. So they do this through transcription factors, which are little proteins that bind to DNA. And often respond to signals, right? So these signals will often either cause this transcription factor to bind to DNA and then allow some sort of output or response, right? And so we think of these like little circuits, right? We have some sort of output, or input, we have some sort of output, and these are mediated by certain proteins. Now, this can be done in a lot of ways. Sometimes these transcription factors turn genes on, sometimes they turn them off, right? Sometimes that signal makes it bind, sometimes it makes it unbind. So there's a lot of complicated parts of this regulation that occur. To simplify it a bit, we normally draw them in these like node diagrams, right? So we have one input, we have this node for the transcription factor, and then the gene it affects, and of course the output that gene alters, right? And this is the simplest case, right? One gene, one transcription factor, one output. We know though in the actual cell, this is not typically what happens. Oftentimes these transcription factors will affect many genes at once. And sometimes all of those genes that they affect will include one response. So this is the case oftentimes for like biosynthetic pathways, right? Where all of these genes encode enzymes that are necessary to produce one amino acid, right? Other times though, each of these subsets of genes are going to control different responses. So we have the first three for response 1, the second three response 2, and the last three for response 3, right? And they can vary what they're doing. What this allows the cell to do is take that single input and disseminate that information into a lot of different phenotypic outputs, right? Maybe you want to alter your metabolism while you're also upregulating stress response genes, right? Doing multiple things at once, right? And truly, again, even this, while more complicated than the original, is quite reduced compared to what actually happens in the cell. Because typically, these sort of networks of transcription factors are even more complicated because they have other transcription factors that can then feed in, right? What this allows the cell to do is create all these sort of conditional expression patterns, right? Maybe you need transcription factors 1 and 2 in order to cause the response. If only one is around, it won't do it. So this allows the cell to create more complex decision-making abilities, right? And I think this is sweet. This is what I've studied since I was in grad school. I love transcription. It's so fun, right? But one of the things that I have since started studying is not just understanding how this happens in one organism, but how it happens in different organisms. And in particular, organisms that have the same transcription factor, right? And that's because if we say that like species X here has the network that we just showed on the previous slide, if we look at other closely related species here labeled as Y and Z, those networks actually might change a bit, right? Some features of those networks will stay the same, so we consider this a sort of conserved response. These responses are often things that are quite ancestral. These are things that existed when this transcription factor was very early and often very highly required for the fitness of the cell, right? In other instances, this will change. There'll be species-specific sort of responses that occur, and sometimes these are adaptations into their environmental niches, right? And this occurs via rewiring of the transcriptional network, where rather than targeting these 3 genes here, they'll target a new set of 3 genes via changes in the DNA, right? In other instances, we might have the same type of response. So all 3 of these are pink, but the intensity will change. So species Y has a really strong response because it's a lot darker. Species Z has a really weak response, and species X is somewhere in the middle, right? So one of the things we really want to study is truly how these sort of networks of transcription factors change across species, right? Because this is super important for us to understand how they're going to adapt into different environmental niches, right? Or understand how they alter their programs in different circumstances, such as pathogenesis, right? The other thing, though, that I like to study, and this is why I brought it up earlier, are phage, right? So these are little viruses that infect bacteria, right? So they're going to infect that cell and they're going to inject their genetic material in. They're going to hijack the cell's machinery in order to replicate. And then they're going to cause that cell to pop or lyse, right? Lysis just means to destroy the inside of the cell and spread that infection around. Basically the same premise as when we get infected by viruses, right? They're going to use our own stuff to make more of themselves. And this is pretty rough for that cell. The cell dies and then potentially kills all of its friends around it, right? Because of this, this has put a lot of evolutionary pressure on bacteria to develop immune systems against these cells, right? For those of you who don't know, actually, a lot of the immune systems that our cells use have homologs in bacteria against viruses, right? So these bacteria have developed immune systems to combat these, but of course, in turn, the phage have often developed ways to counteract those immune systems. So they're really locked in this evolutionary arms race between one another, right? And this is sort of fought on several fronts. I'm going to use some analogies here, right? Sometimes this is direct combat. And what I mean by that is that the 2 organisms are directly trying to destroy one another. So oftentimes, for any of you who have heard of CRISPR, right, which has come up a lot in the news and stuff, we use it to edit human genomes. Turns out that's actually a bacterial immune system that was identified, right? And what it does is it targets very specific sequences of DNA. And it's actually the first adaptive immune system that we have found in bacteria, right? Because it can adapt to different infections, right? And so this will cut up the bacterial or the viral DNA and try to basically stop the infection from progressing. And in turn, phage have often created other proteins to sort of counteract these CRISPRs, and we refer to them as anti-CRISPRs, not very original, but effective to name, right? But there's also like anti-CRISPRs which try to go against this. So there's sort of this constant back and forth between the two. Now, other times though, that this battle isn't fought so directly. It's fought in what I consider to be more espionage, like classic spy movie stuff, right? So essentially what we will see is that sometimes viruses will try to manipulate their hosts in order to make them a better environment, right? Oftentimes we see this in reference to the host metabolism. So cells are going to upregulate the biosynthetic pathways that they need to either go through protein synthesis, because when the virus is going to infect, it's going to need to make a lot of proteins to replicate itself, right? Or certain energy metabolism. We often see this present in a lot of cyanobacterial phage. Cyanobacteria are the ones in the water, they're going to do a lot of photosynthesis. And so many viruses that infect them will often contain the photosystems that they use for this, right? And that's cool. I don't really care about it that much though. What I care about is actually when viruses use transcription to do this, right? Something that I refer to as transcriptional espionage, where basically viruses will encode factors in order to manipulate their host's transcriptional network, right? And what they can do for this is they can do a lot of things. Sometimes this can help them regulate host metabolism. Sometimes though, this can help them just regulate what behaviors the cell is going through. And this is something I love because it puts both of these things together, phage and transcriptional networks. And you might be wondering, why am I studying this in the first place? Most people do, and that's fine. It's good to justify what you're doing and why people spend money on you. The big reason is because phage are an incredible therapeutic. So turns out you can use phage as a therapy. It's referred to as phage therapy, not very original, but fun, right? So what can happen is if you get phage that target certain infections, right, certain bacteria that are causing these infections, you can effectively target them in a patient who is suffering from an illness, right? And this is actually slightly better than antibiotics, because antibiotics are very broad-reaching. Antibiotics will destroy all of the microbes that they attack, whereas these phage are often very specific. They'll only target 1 or 2 bacteria. Okay. So this allows you to have much more precise treatments. But the big reason that we have started to look at phage as therapeutics is because of the healthcare industry. You will be unsurprised to know that they have misused a lot of stuff. And one of the things that they have misused over time is antibiotics, right? And the misuse of antibiotics has caused a lot of antibiotic resistance to appear, right? And so we need alternatives to antibiotics because of this. You might be asking, why don't we just develop more antibiotics? Well, that is because antibiotics are costly. It actually costs companies about 500 million to 600 million to actually develop and research new antibiotics, and they don't typically profit until 23 years later. And so a lot of pharmaceutical companies just don't care because it doesn't line their pockets, right? And so we need these alternative strategies, right? And so phage have become a really hot topic in the last decade or so. And you might be thinking, oh, why is it just in the last decade that we have thought of this? It's in fact not. This phage therapy is actually over 100 years old. The reason, though, it was largely abandoned right around the 40s to the 60s. This was because we identified antibiotics right around that time, and companies thought this is way easier to use, but it's also because of politics. A lot of the phage therapy and the studies that were being done about them were done in Eastern Europe and the former Soviet Union. And so when Western scientists figured this out, they decided to abandon a lot of that for political reasons, right? And so this has gained a lot of attention. And this might sound very similar. In 2025, you're also seeing a lot of good old government sanctioned removal of science for political agendas. So history repeats itself several times, right? And so we want to study how these two things interact, right? How do phage infect bacteria, right? And of course, how do bacteria respond to this? And if we can understand this, we can design better phage to be therapeutics, right? We can design them with new transcription factors or new molecules to regulate their host to kill them better. They can be better in the patient, right? And actually, I mentioned this before, this strategy has been used really effectively for a lot of patients who have multi-drug resistant infections. There's a company from a professor down in, I want to say it was Harvard, that he had started this, but it's actually, I find it to be quite scary. He gets these patients and like within like a day they have to have all the stuff ready, because otherwise these patients might die. Right? Really intense stuff. Right? I don't work on anything that intense. I work on very chill stuff. I promise. So to study this, I use the bacteria Caulibacter crescentus. Caulibacter is this lovely little thing over in the corner here, right? It's dimorphic, so it's got one cell that has a stalk. This is the stalk, and one cell that has a little flagellum. Flagellum are what they use to swim, right? And basically, this is a parent cell, and every cell it creates will have a little flagellum, and then it will swim away and eventually find its own little area to live and create a stalk and stick there, right? And they do this via what's known as the holdfast. So this is an adhesion produced at the end of the stalk, and it actually is like anywhere between 3 and 8 times as strong as superglue. And so they will irreversibly stick to a surface and form what are known as biofilms, right? These are basically conglomerates of cells stuck to each other, stuck to surfaces, and often coated in some sort of extracellular polymeric substance. Sometimes sugars, sometimes DNA, sometimes proteins, sometimes all of them, right? And so it's really cool, but it's also a member of what we refer to as the alpha proteobacteria. It's a broad group of bacteria that you've probably never heard about. Don't worry, you don't have to know anything about them. We'll talk about them a little bit later, but this will come on, right? So we have our bacteria. What's the virus that we're going to use to study this? It's known as Phage. So this is the lovely little image of Phage. So up here is the head and down here is the tail, right? And this over on the right-hand side is actually a cryo-EM image of several of these phage infecting the cell, right? So here's the host cell, and all of these are little viruses. So you can see their little tails attached to the end where they're injecting DNA, right? What's cool about Phage is that it's actually really well characterized. It was used as a workhorse to study Caulibacter biology for reasons I won't bore you with. But what's useful for me is that it was really understudied in terms of what it did, right? We knew how it affected Caulibacter, but we didn't really know much about what it was doing during the infection. So it's sort of like a gold mine of areas to work with, right? So we wanted to study this. And so we had to study, of course, the transcriptional regulatory networks in these species, had to start somewhere. And I did that with protein known as an XRE. So these are just referred to XREs. These are transcription factors that have a particular domain. Be unsurprised to know that that domain is called an XRE domain, which is shown up here in pink, right? This is the area where it actually binds to DNA, right? And here is just an AlphaFold structure, or the predicted structure of it, where the DNA binding portion is going to be right over here. They have this funny little loop here, which we think might be actually allowing them to interact with other proteins. Right. What was cool about these XREs, though, was that Caulibacter didn't just have one of them, it actually has 4 copies, right? And each of these copies looks highly similar to one another. So this is a multiple sequence alignment. So the darker the color, the more similar it is between them. And what you can see is that particularly in that XRE region, all of these species are quite dark. So they're almost identical, right? I think they're between 80% identical between one another, right? And we actually think that these are paralogs of one another for reasons I can show later if you care. But I'm not going to talk about right now. And so we wanted to know how these XREs were affecting Caulibacter, right? And to do this, actually, not yet. One of the reasons we care about these XREs, got a little ahead of myself, is because they're not just present in Caulibacter, they're actually present in all of the alpha proteobacteria except for one group, right? And they're highly conserved even at the level of genomic position. And a lot of these species in the alpha proteobacteria are very clinically and ecologically relevant. We have things like Brucella, Bartonella, and Rickettsia, which are all pathogens. Brucella abortus in particular causes abortions in cattle and in humans, right? Bartonella and Rickettsia causes rickets, right? There's plant pathogens like Agrobacterium, which causes crown gall disease, which is actually kind of fun to work with. But the other thing is they work with a lot of plant symbionts. So these are things like Cynorhizobium, Rhizobium, Leguminosarum, which are actually able to promote plant growth. And so we can use them agriculturally to enhance crop yields, right? And so one big question was, what are these XREs doing, right? And why are they so highly conserved across all these species, okay? So we had to take a stepping stone, and we decided to stick with Caulibacter at first, and then start to analyze the other ones as we went along. Today, we're just going to talk about Caulibacter's ones because it turns out there's an interesting story there. So to analyze what these XREs were doing, I typically take big omics approaches, right? So these are big data approaches where we're looking, for instance, at all of the binding sites for these XREs. Where are they binding in the genome, or what genes are they regulating, So this first one here is a ChIP-seq plot. Okay, for those who don't know, ChIP-seq is essentially an assay that we use to ask where's a protein bind to it, right? And what this plot is showing is on the x-axis, we're looking at the position across Caulibacter's genome, okay? And on the y-axis, we're looking at enrichment. And essentially all you need to know is that each of these individual peaks is a site within the genome where these transcription factors can bind. So we're able to map all of the different sites that they bind to. Turns out that they bind to hundreds of sites. In one case, I think there's over 1,200 sites. This is atypical for a lot of transcription factors. Most of them only bind to a couple, right? And we're able to do this not just for one of the transcription factors, but all 4 of them. And what we're able to see was that all 4 of these transcription factors have very similar patterns. It's hard to see at this big scale, but I think anywhere between 60 to 99% of those sites overlap with each other. So they largely have redundant functions together, which is kind of cool. But we didn't just want to know where they could bind. We wanted to know what they were actually regulating, what genes were being changed when we used them. So we combined this with a protocol or a assay that's known as RNA-seq, right? RNA-seq is essentially looking at all of the RNA in the cell at once, right? And what we do is we do this in 2 different mutants or 2 different strains and compare them to each other. See how the RNA changes in one versus the other. And in particular, we did it in a wild type, which is labeled as WT, right? And a mutant in which we deleted all 4 of the XREs. And basically just asked, how do genes change when you get rid of them, right? And then we can use this sort of to understand what genes they are regulating, right? So on the x-axis, we're looking at the log2 fold change between these 2 strains. And anything above 0 is going to be upregulated when they were deleted. Anything below 0 means that they were downregulated or turned off. Right? And what you can see is that most of the genes were upregulated when you removed these genes. And with some sort of logic here, it's a little sometimes backwards because it's double negatives. Because we removed these genes and they went up, these were transcription factors were actually largely acting as repressors. They were turning those genes off. Right? So when we removed them, they were able to turn back on. Right? And what we're able to do is then combine this RNA-seq with that ChIP-seq data and ask, well, of the genes that got turned on, how many of them had binding sites for those transcription factors in their promoters? Promoters are just basically the regions that control when they go on. Right? And so we can ask which ones are direct, and those are just shown here with these little red dots. Right? So I think there was about 100 here that met our statistical cutoff for genes that were upregulated and had direct binding sites, right? Some of them you'll see here are blue. So these are sites that were significantly upregulated, but didn't have a binding site for the XREs. And these are likely through some sort of indirect method, right? You can imagine a situation where one of these red dots encodes for a different transcription factor that when it gets upregulated, turns something else on. So those are likely through indirect secondary steps. All right. And this is super cool, right? We get to know all the stuff. We get to map the whole transcriptome for this. But what does this actually mean for the cell, right? We're going to take this and look at some physiology based off of these, based off of this data. In particular, look at some of the genes that were being upregulated over here. And so 2 of the genes that we cared about were HFIA. HFIA is an inhibitor of holdfast formation. Remember, holdfast are those little sticky substances. And so by acting as a repressor of HFIA, these XREs were actually able to enhance the holdfast, right? So they repressed the repressor and therefore they enhanced it. A lot of double negative logic here, which I have a horrible time with, so I have to write the arrows, right? And this is super cool. So these cells, these systems were allowing the cell to control when it was adhering to surfaces, right? But we also saw that it was able to regulate when the cell was going to lyse. So there are certain programs in the cell that will cause it to basically pop prematurely and do some weird biology that I'm not going to talk about today. Well, what we saw was that if we overexpress these XREs, they inhibit this GAF-YC, which of course which promotes cell lysis. Because it's inhibiting the thing that promotes cell lysis, it actually clamps down on cell lysis, right? So we're able to take this big data, narrow it down to some physiology, and then ask some questions that way, right? And of course, there's a lot of other genes here, so we can do a lot more interesting stuff to see what are the other pathways that are being influenced. But it turns out that there were some interesting bits here because it wasn't just Caulibacter that had copies of these XREs. By accident, I found that by CBK, that phage that infects Caulibacter actually encodes its own copy of these transcription factors, right? And so here over on the left-hand side is just another multiple sequence alignment, where we're comparing the 4 host XREs to the phage XRE. And you can see particularly in that DNA binding region, they're as similar to the host proteins as the host proteins are to each other. And we think that the phage may have co-opted this transcription factor for its own purposes. And so we wanted to start testing this, right? Understanding why is it that the phage has something that looks just like the host. And so we had 2 broad ideas of what this might be doing, right? The phage is going to infect Caulobacter, and then it's going to express this XRE, right? And we thought that this XRE might be first impacting host transcription itself, right? We know that the host factors they affect a lot of things based off our RNA-seq. So maybe this phage is using this system to alter the host transcriptional profile. And that maybe by altering it, it's able to enhance its own infective abilities. So to test this, we wanted to first look at its ability to alter host transcription, right? And just going to go back to the same techniques. They worked with the Caulobacter XREs. They're going to work with phage one. All right. So we're able to do the same techniques. We took the phage XRE and we basically just added it to the host by itself away from the phage infection, right? And just asked where did it bind in the host genome, okay? And so we did ChIP-seq again. And so again, we're just looking at on the x-axis, the position in Caulobacter's genome, the y-axis is the enrichment. So what we're looking at is the different sites that this Caulobacter phage protein is binding, right? And so it binds to a lot of sites, and it turns out that 99% of them overlap with the host sites. So the things that it's going to regulate are almost entirely redundant with its hosts, XREs. And we can confirm this by doing RNA-seq. So we overexpressed this XRE in Caulobacter and asked what genes was it altering or regulating, right? So what you'll see is that when we overexpress this protein, most of the genes are being downregulated. So anything below 0 is going to be things that are downregulated or repressed or off, right? You'll notice that this looks slightly different from the last RNA-seq where everything went up. And this is because in that case, we were deleting genes and asking how they changed. In this case, we added the phage gene to this and asked how it's changed. So they have basically the opposite direction, right? So most of the genes were being repressed, just like was consistent with our XREs from the host. And amongst those were genes that we had identified before, right? HFIA and GAFYC. So what we were able to ask is, hey, you see that these are being transcriptionally regulated. Are these affecting the phenotypes that these 2 genes are involved in? So we could then overexpress that XRE from the phage in Caulobacter and ask, so we were able to ask what happens when you overexpress this. And essentially what we saw was consistent with Caulobacter. If you overexpress the phage protein, HFIA expression and GAFYZ go down, right? So it represses both of them. And consistently, the phenotypes match. So we could see that when you repress it, adhesion went up and cell lysis went down, right? And so in fact, this phage XRE could alter the host transcriptional profile. But of course, the other big question was why? What benefit does this transcription factor have for the phage other than making it a little stickier? Was it able to alter the infection in any way? And of course, this also led to the other idea, is if the phage transcription factor can affect infections, what were the host transcription factors doing in infection? Did they play any part? Why is it basically hijacking this host system? So what we did was use an assay where we measured basically what we refer to as the burst size. So the burst size are the number of phage that are produced per infected cell. The reason we call this is because when the cells lyse, they pop, and those phage are shot everywhere. So it's essentially quite literally bursting out them. And so we can measure the number of phage that are being produced per infected cell in strains where we have removed those XREs and added them back and see how does it affect those numbers, right? And we can see here, this is the burst size in either a wild type, which is again is just abbreviated as WT, or a mutant in which we deleted all 4 of the host XREs. And what you can see is that the burst size decreased by a factor of 2. So half as many phage are being produced per infected cell. And if we add back any of the individual XREs, shown here in pink from the host, or the XRE from the phage in blue, we can see that it restores that phenotype. So if you overexpress these XREs, we produce more phage. If you remove the host XREs, you produce less phage. So it's actually a little funny because the host XREs are somewhat deleterious for the host, right? We'll talk a little bit of why that might be in a second, right? So based off this, it does look indeed like these transcription factors not only impact host transcription, but they do seem to enhance their own infection, right? So I want to put this together in sort of a model for you on how we're starting to think about this, right? We have Caulobacter that has these cool XREs, we've got 4 of them, right? They infect a whole bunch of genes from the host. Some of those genes regulate adhesion, right, by inhibiting H5A. Some regulate cell lysis by inhibiting FYZ. Others, though, seem to actually promote phage production, right? Because if we remove these, phage goes down, right? And so what that suggests is that in the presence of them, they're actually making the phage more able to replicate. And what we think that these XREs are doing in the native host isn't making them worse when they're around phage. It's that it's protecting the host from phage defenses, right? That they act as inhibitors of phage defense. You might be wondering, well, why would you do that? Clearly, it's not good when you're being infected by phage. Indeed, it is not good when you're being infected by phage to have those defenses down. But every other time in your life, having those up can be really deleterious, right? Think about the ways that we have autoimmune disorders, right? When your immune system is overactive, it has a really negative effect on your health. And the same thing goes for bacteria. If you let these systems run rampant, they can actually be really deleterious for the growth and fitness of the cells. And so we think that this is a native system that the host uses to sort of clamp down on phage infection, or phage defense proteins. And that we have 5-CBK has its own copy, does the same things, and in fact does this in order to enhance this infection by basically keeping those phage defense genes off, right? So the basic idea here is that it more or less hijacks its host system that it's already using to control phage defense in order to maintain it, right? And this is super fun. Caulobacter is lovely. I love Caulobacter. This is all work I've done though in the past, and I want to take this work out elsewhere. And this is what I'm going to be, what I've started doing as a faculty member. It's not just looking in Caulobacter, but all of these other species, right? We have Brucella, we have Bartonella. I'm not going to work with Bartonella. Bartonella's scary, right? But we have Rhizobium, Agrobacterium, all these different plant symbionts and pathogens that are clinically and ecologically relevant. And what we can start to ask is, how do the XREs in these other species look? Do all of them control adhesion? Are they all regulating phage defense? What are they doing in these other species, right? What's really lovely about this is when we do this, right, we're able to sort of compare how that network changes. But each time we do this, we're learning sort of 2 things at once, right? We learn how that network changes, but we also learn about what that network is in a new species and all of the potential things it might regulate, right? We know, for instance, in, which one is it? Rhizobium lignosorum, that the XRE there actually promotes adhesion, which allows it to be a better nodule form. I think it's right, Rhizobium, right? And so we sort of get 2 bits of information. We get to understand the evolution of them, but we also get to understand what they're doing with these other species that might be really important for us, right? But we're not just interested in how they're doing this in the bacteria, right? But also in the phage, because it turns out that a lot of alpha proteobacteria have phage that encode copies of these XREs, suggesting that many of them have co-opted them for some sort of use, whether that be to promote their own infection or something else, we don't know yet. So one of the things we want to do is basically take this research and compare it in these different species. Take Agrobacterium and its phage that has the XRE and compare what each is doing in each of them. And basically go through this thing where we map those networks using ChIP-seq, RNA-seq, these kinds of things, compare those networks, predict phenotypes, and of course test. You can do this on the host side and on the viral side and start to better understand how this is happening. If we can better understand this, we can sort of start to build better phage, right? Rather than provide them with molecules that combat host defenses, provide them with molecules that make sure the host defenses never turn on in the first place, right? And I actually have 2 undergrads working in the lab right now. Right now, we can of course do this while we're getting set up. Some of this might be hard to do at the web bench work, right? But we can actually do this on the computer as well. So when we do ChIP-seq, you're actually able to get all of those, 1,200 sites, and actually identify sequences that are enriched in those sites. And these are sequences that they likely bind. And so this is the binding site for XRE1. And what we can do is then have students go through and take new genomes for other alphas that have that speed, have that transcription factor, and predict where they're going to bind. So we can already have targets in mind, the things that we care about or we're going to look at, right? And so McKenna Fisher, who's over there, say hi, McKenna. And Charlotte Barron are going to start working on this. McKenna's already started, right? And so this is super fun, right? We can do a lot of stuff with this, but this, of course, is going to be largely focused on things that we already have in the lab, right? What about new things? Well, one of the things that we can start to do on top of this is look for new phage, right? And we can look for them pretty much anywhere. Like I said before, they're very abundant, right? And so in my postdoc, some of the folks in the lab were doing work at Lake Lansing, right? I did my postdoc over at Michigan State. This was like 5 minutes down the road, right? They would go over there and collect water samples and they'd grow bacteria. It turns out, by accident, we identified a phage from Lake Lansing, right? And so what this is this is a plate here, right? And all that cloudiness is bacteria, right? So the bacteria are able to grow in this big, what we can call a lawn, right? And when you add sample from the water, the lake water, it forms these little areas of clearness. These are called plaques. These are essentially areas where the phage has killed all the bacteria. And so it isn't able to grow and make a big cloud. And so we identified a bunch of these plaques from Lake Lansing, right? And we were able to do really fun stuff with this, right? I was able to take this, isolate the genome, assemble it, annotate the whole genome, right? In a matter of a couple days, right? And what we found was that this is actually a new brand of Calybacter phage. This was what we refer to as an N4 phage. They're present in they have other hosts, but we've never identified one that infects Calybacter before. And so we have this new N4 phage, and then we were able to start characterizing what this phage does, not just what it looks like at the genome level, right? So one of the things we looked at, of course, was how well does it bind to Calybacter? So you do what's called an absorption assay, where essentially what we look at is at the different time points, we look at the amount of phage that are stuck to a Calybacter, the amount of phage that are floating around, right? And we're able to do these absorption curves where we're plotting the percent unabsorbed, and you can see that by 60 minutes, almost all of the phage have bound up to Calybacter, right? We're also able to see how well it kills Calybacter, right? So this is what I would call a clearing curve. McKenna has seen this in my micro class, right? Where basically we add phage to cultures and see how they grow over time, right? And so on the x-axis, again, we have time. On the y-axis, we have optical density. This is essentially just how cloudy the cultures are. And the cloudier they are, the more cells that are there, right? And so you can see that in the 0 MOI, the black one, MOI stands for multiplicity of infection, which means we just 0 is no phage, right? They grow just fine. You can see that they grow until about 10 hours or something, and they sort of plateau out because they've reached their carrying capacity. If we add either 0.1 MOI, which is essentially less like 1 phage for every 10 cells, or 1 MOI, which is essentially 1 phage per cell estimates, right? We can see that those cells will grow for a bit of time before the OD starts to collapse. And this is because those phage are completely lysing all the cells present. And eventually you get down here where the only thing left is all the debris from inside the cell, right? And so you can see that they're able to clear them quite well. They're able to kill these cells off, which is super fun and super cool. Right, but we're able to even take this a step further and start asking ourselves, well, what genes in Calybacter are important for this infection? By doing what's known as bar-seq, which is basically transposon libraries using randomly barcoded transposons. That sounds really fancy. I promise you it's not. Essentially, all this is, our transposons are jumping genes, so they're just going to jump inside of the middle of another gene. And when they do that, they're going to destroy that gene, typically. And each of the transposons has its own little DNA barcode that we know where it's from. So what we do is we can map a whole pool of these. So we might have 100,000 mutants in there affecting all the different genes of Calybacter. And we can basically then infect them and ask, well, which of those are able to survive, right? And we do this by essentially amplifying the barcodes and counting them, right? And so things that are able to survive or are enriched are going to have more barcodes at the end and have what we call a higher fitness score, whereas ones that aren't able to survive are going to have less barcodes at the end and have a lower fitness score. And so we did this when we infected cells. We basically did a killing curve. And at these different time points, just measured the number of barcodes we saw, right? And that's over here. And on the right side is actually what we draw as this like fitness plot. So on the x-axis, we have the times post-infection, right? You can see this arrow here matches with this sample right here. So right at the very end, once we cleared the infection, what we basically looked for was strains that had a high fitness at that point when all of the things were dead. Basically, what was still growing at the very end. Right? And each of these different lines is a different gene. So it's a different gene that was knocked out by one of the transposons. And so each of these genes, when you disrupt them, actually allow the cell to grow better. Right? And it turns out that most of those genes are involved in production of what's known as LPS. LPS is the surface molecule on top Caulobacter. It's on the outside of any gram-negative, right? And we see that if we disrupt any of these, which are shown here in pink, those cells are much happier. And what we think is that the LPS is acting as the receptor for the phage, right? And so by removing the LPS, there's nothing for the phage to bind to anymore. And therefore, it can't infect those cells, right? And this is all fun and cool. This is a fun little project, but we don't just want to do this with one phage. We actually wanted to implement this into a class. So me and Emily Labring are starting a C-Phages program, which is partnered with HHMI, who's run this program for a long time, right? Where essentially we're going to be having students do this exact thing, right? So they're going to be able to go out into their environment, take samples, identify phage that infect and kill stuff, isolate them, purify them, do the genetics, do all the bioinformatics to annotate them, and of course characterize them and stuff like that. And so we're super excited about this. This should start next year. I don't remember. Okay, there we go. And so with that, I'd like to thank you all. I only have a picture of McKenna because it was really short notice. And I think it was from Charlotte. But if you have any questions, I'd love to take. Yes.
Curious about the population dynamics. In the lake, you have the slide, the florey stuffing bacteria.
Yes.
And the phage. Why haven't the phages just wiped out all of them?
Yeah. So it turns out these phage were actually quite hard to get from the lake. And the reason being was Caulobacter isn't super present in the lake. And so what ended up happening was because of the way that the experiment was run, my boss was basically growing Caulobacter in lake water for a long time. And then what ends up happening is eventually they get enriched. And so we think that there was maybe only one phage particle in their original sample, which is like 4 liters. And then eventually got enriched because of the way they were doing it. The way that we think about this in terms of like population dynamics is oftentimes we see resistant mutants start to pop up. And there's some sort of balance there of like basically keeping those low while the phage, because of the phage infection, of course, not everything can do that because a lot of LPS mutants are quite sick. And we know that because like, if you look here, most of these mutants are actually doing really bad at the start.
Yeah.
But the other thing is that there's a lot of defense systems that allow Caulobacter to persist. One of the best defenses is actually that cells kill themselves as soon as they get infected, so that they don't spread their infection around. And so we call them abortive infection models, right? So they often basically use toxins to kill themselves. And so there's a balance there of basically maintaining your population using defenses while not overusing them. Causing utility. Yeah.
There's a question from a student on.
Okay.
Just gonna read this verbatim.
Yes.
I don't have a lot of knowledge in biology, but my
I do.
question is related to how fast phage evolve. Can they evolve during its administration as a therapeutic? Could that decrease the phage's effectiveness or make it deleterious to patients?
Yes, it in fact can. And less to do with whether or not the phage evolves, but whether or not the bacteria evolves. Because the thing is, the phage isn't going to cause any problems in the human typically. Weirdly enough, it turns out human cells will actually just kind of uptake phage and then I think destroy them. Those for sources. But what can happen is that, sort of like you see over here, right? Sometimes random mutations occur and the bacteria will lose whatever receptor it's using, whatever receptor the phage is using, and so it becomes resistant to phage infection. And that, of course, would be terrible because you're not able to clear the infection as well. This is why there's 2 things that folks have done to sort of combat this. They often use combination therapies of a phage and an antibiotic to hit both targets. The idea is like, if one is unlikely, right, if you have the unlikely event of one mutant being resistant to the phage, it's also unlikely that they would have a second mutation that would make it resistant to the antibiotic. The other thing though is sometimes folks don't care so much because mutants that lack the receptors for phage often are less virulent. So a lot of the phage receptors are things for like pili and flagellin, which allow for the bacterial to latch onto its host or swim to where it needs to be. And so by removing those structures, it makes them resistant to phage, but less pathogenic. So there's sort of a weird balance there of like, they become phage resistant. Sometimes that's not actually that bad of an outcome. Not always, sometimes.
So I saw an article about engineered versus non-engineered phage therapy. I didn't actually read yours, so I'm gonna ask you.
Yes.
So what is engineered therapy? Is it, I mean, is that like, you know, you go to the doctor and you get multiple antibiotics, you need this phage and this phage, or is it you're actually manipulating the phages in a way to make those therapies better for the particular infection that they're trying?
This is a great question. I don't know what the article in particular is referencing. We can sort of do both.
Okay,
So one of the things that they do for like the guy who has the company, what they do is they have to get a culture from the patient and identify what the species is that's infecting the patient, and then identify what phage they have to kill. And so you do need a specific phage. You can't just give it any. So you do need a specific one there. But you can, of course, start to engineer these phage with additional things. Like one of the issues, right, is the toxin antitoxins that cause the cells to self-kill themselves, right? Sort of redundant, right? Oftentimes they have an antitoxin, so they don't do this all the time. There are certain phage that encode those antitoxins to prevent that. And so, you know, one of the things is, of course, you could engineer the phage to have basically counters to those host defenses. The genetics on phage can be very difficult, depending on the type of phage, but some are very viable for the genetic.
Okay. Kind of related, but I was trying to, so like you can go like fishing for
Yeah.
phage kind of, right? Like,
absolutely.
in the natural system, these ones that could potentially be therapeutic for infections and
Yes.
bacteria that are like human.
Yes.
Like, how are those ones found? Was it like cultured, like someone, and then they're like, oh, this isn't growing, there must be something going on, or like.
Yes.
So is
Sometimes.
that a way to find more of them, is like just
Absolutely.
start getting people to send in their.
Absolutely. It's actually, it's the funny thing, sort of back to Cam's question of like, how is the phage not overtaken the lake? Caulibacter's not good. Right? Where you can find a lot of Caulibacter is at a lot of like waste treatment plants or a lot of paper mills, right? They love to break down lignin and a lot of like cellulose products.
Yeah.
So you find a lot of Caulibacter there. Turns out you also find a lot of phage there. So wherever you are, your killers are also kind of close behind you. So the best places to look for these types of things is wherever we find them. So for a lot of E. coli phage, all you have to do is go to a septic tank. Go to take some wastewater. And they are plentiful. Because they're gonna kill off, they're gonna basically only be prevalent around where they're gonna go. And so like you can always take it. And some of them we've just found was like by accident. Sort of like this, or like, I mean, one of the things that people often fear is actually like, if you work in a micro lab, having phage infections can actually ruin your lab. Because they'll like just destroy all of your cultures. Particularly like E. coli phage, they'll like ruin E. coli culture in like an hour. You'll just watch it like plummet to nothing. It's very sad.
My other one's
Yes.
weird.
Take it.
So like we get into this discussion quite a bit in classes,
Mhm.
I think. So I study things at community level. I know like evolution, like natural selection, we train students, this happens at the population
Yeah.
level. I'm wondering, from your perspective, for someone who studies like the parts of the pieces that do the thing and seeing how these transcription factors alone are like conserved,
Yes.
and they do these things, like from your perspective, like at what level does natural selection occur?
I think it's at the population level.
Still the individual organism, not
Yes.
at like oh, some weird like selfish gene, like I
No,
like that. I
I-
don't
yeah.
know.
Yeah, I think For instance, like for the conservation across the different microbial species, what we think is that that gene is ancestral. That whatever like universal or last common ancestor of the alphas have it. And that everything subsequently has kept it around. Part of that we think is because the XRE1 is sort of the one that's conserved across everything. And it's because it's around a location that is super essential, right? And so it's hard to remove that piece because everything around it is essential. And so it's hard to sort of lose it. And so we think that's at least that. What we think potentially from the phage one, we have no idea. Like if I had to speculate, I would speculate that they probably acquired those genes from their hosts. Like quite directly. Like we don't think they de novo have evolved that to be that close looking to the host. We think that during the infection, like there's a lot of DNA swapping that can happen, or a lot of mutations that can occur, or like recombination that can occur. And so we think that during that, it may have just acquired that from a host that it could infect. And that we often see this, and they refer to them when they're metabolic genes, they're called auxiliary metabolic genes that are essentially host-derived things. We think of these as sort of auxiliary transcription factors that they've acquired from their hosts. And so we think those might be acquired from the host at some point and then maintained the population.
I was thinking like, it's like they're the teeniest, tiniest symbionts in the world, like
Yes.
a lot, yeah.
Yeah, and actually, these are only a particular kind of phage. There are phage that, these are lytic phage, so they always cause lysis. There's other phage that are lysogenic. These are phage that will incorporate into their hosts and actually just sit in their genomes. And then eventually when the host is in a bad situation, they'll pop out and kill the host with the thought that like, basically they're going for a free ride until things are bad and then they're gonna murder the host and leave.
I was just kind of curious, when you were talking about how one of the ideas for why the XREs work the way they do is that they, under normal circumstances, they downregulate the defenses. So do you see activity where they upregulate the defenses if they're affected by a phage that does not have the Phage CDK?
Oh, this is what I wanted to do so bad.
Oh, okay, all right.
I've wanted this so bad because folks have asked, they're like, well, why don't you see, this changing with the Phage CDK? And I was like, the problem is, like when we do these infections, like you mentioned, like right here, right? When you infect this wild type or this XRE mutant, the phage has the XRE. So anything that would be turned on potentially would still be turned down because it's also, I didn't show this, but the phage XRE is one of the highest upregulated genes at like 10 minutes post-infection. Or 15 minutes post-infection. So it's very early and we think it's basically setting up the host for this. And so we would love this. And one of our hopes was to delete the phage gene to see like, do we see this suddenly changing or being different? Or like, is there a much more drastic effect here? We haven't been able to do it yet.
Okay, no, I was just curious.
It's the perfect question. The reviewers asked us the same thing.
Then I have an unrelated.
Yes, absolutely.
About the XREs. So this is just because you brought it up, I'm kind of curious. You said that they look like they're paralogs.
Yes.
Like it was. Did it affect how you did the analysis?
Yeah, so
The original.
we think that they're paralogs because if we look at their similarity, they cluster together. So there's like, these aren't the only XRE containing proteins. I think there's 30 of them in Caulibacter. And so if you compare them, they basically cluster off into these 4 that look highly similar to each other, and then everything else that doesn't look like them. And when
the other XREs work the same way, I was curious.
They do.
I've never heard of it, so.
Yes, so as far as we can tell, we don't know if they bind to any ligands to alter their functionality. That's always an easy one, like what signal is causing them to do this. But it has made things quite difficult because to see some effects, you have to delete all of them. It's a pain to delete 4 things. And actually, my favorite one, I have it here, is that, there's various numbers of paralogs in other species, like this magnetospirillum, which is a magnetic bacteria. It's super cool. Yeah, it's got a bunch of magnetic crystals in it. Has about 8 paralogs. There's a Caulibacter species that has about 12 of them. And so like there's some really cool stuff, but it's also like balancing, like, I don't want to delete 12 things. That's a lot of work. So it is been an interesting thing of like, why have they done this so much? And is there any sort of like neo-functionalization or sub-functionalization between them? It's been hard to say with the callback words, like. It's also asking like, what was the limit of binding in our ChIP-seq data? Like, are the places where they're not binding real or not? Or is that just because it was slightly weaker in that one? Hard to say. Thank you so much for coming.